Hello and welcome to this glossary of different terms used in genomic medicine or molecular medicine. 1. Allele. One of the alternative forms of the gene occupying a given locus on the same chromosome. Antiparallel or antisense, where an RNA molecule is a mirror image of the RNA, because of which the antiparallel RNA can hybridize with mRNA and prevent its translation. Autosomal inheritance. It is an inheritance which is not sex-linked base pairs. The Watson-Crick hypothesis that A pairs with T and C with G in the DNA helix. Chiasma is a crossover of the chromosal sites, the place where the two chromosomes cross over. Chromatin, organized DNA structure with histones. Dark condensed chromatin, not transcribed, is called heterochromatin. Chromosome, genetic unit in the nucleus containing DNA-based information for phenotype expression. There are 22 pairs of autosomes and 2 sex chromosomes. Clone, a totally identical cell in genetic composition and derived from a single common ancestor. Coding, the genetic code existing as triplets derived from the 4 bases in DNA A, C, G and T. The code decides the mRNA sequence and the sequence of linking of specific amino acids when a protein is under assembly. A triplet, example GUU, codes for valine and is called a codon. DNA, a dimer in double-stranded helical form consisting of 4 nucleotides A, C, G and T. cDNA, that is complementary DNA. This is obtained by reverse transcription from mRNA. Diploid, a cell containing 2 copies of each chromosome in contrast to a haploid cell where only 1 copy exists. Dominant, when the phenotype is expressed in a heterozygous person, this is in contrast to recessive where phenotype expression occurs only in homozygotes. Eukaryotic, a higher form of cell life in which nuclei, nuclear enzymes and mitotic activity is manifest. In lower cell forms, prokaryotes, such an organization does not occur. Exon, that part of the DNA sequence which is transcribed. Gene, the total nucleic acid sequence needed to be transcribed into mRNA and then translated into a full peptide. Genetic distance, the distance between two linked nucleotide sequences. The genetic unit of distance is a centimorgan. Genotype, DNA sequence constituting the genetic profile. Germinal cells, they are also known as gametes. Mutations can occur at this level. Hereditary disorders, a disorder caused by a mutation in a gene which is inherited. HLA locus, this is a small segment of single chromosome containing many genes coding for functions concerned with immune response. Intron, total blockage of a particular gene expression by a specialized gene transfer technique in laboratory animals. Linkage, alleles which are close to each other and influence inheritance in a combined fashion. LOD score, logs-odds ratio, a measure of the linkage of 2 loci at a given recombinant frequency. Meiosis, germinal cell division by reduction which results in half the number of the base chromosomes. mRNA, messenger RNA, the RNA transcript that carries a specific message for translation of a particular protein. Missense mutation, a mutation in which a codon sequence is so altered as to produce a different amino acid. Mutations may also be of the nonsense variety in which no defined product is formed. Northern blot, the study of mRNA by electrophoresis separation and hybridization with specific probes. Oligonucleotide oligomer, a single strand made of nucleotides in specific sequences. Example, 17 nucleotides strung together in a particular sequence is called a 17-mer. Palindrome, two sequences which are identical if read left to right in one strand and right to left in the other. PCR, polymerase chain reaction, a method by which DNA sequence can be replicated by using thermal unwinding of the DNA helix and then ensuring transcription with TAQ polymerase. Repeated PCR can amplify small amounts of DNA a million times. PCR penetrance is a specific penetrance, a specific genotype which ultimately finds its expression in the phenotype. Phenotype, observed or manifest genetic characteristics. Plasmid, a loose extrachromosomal DNA circle replicating autonomously. Polymorphism, allelic variations in DNA due to change of base in the triplet code. When DNA is broken down with restriction endonucleases, polymorphism may be noted at the break site. Restriction fragment length polymorphism, RSLP. As at this site, tandem repeats of a given sequence may also be noted. By using electrophoresis, restriction maps can be constructed of polymorphic variations. Recombinant DNA, the construction of a novel DNA molecule by joining sequences from different sources. Restriction, these recognize and cut the DNA strand at the end of a designated sequence. Example, HA-AE3 enzyme cuts out every GGCC sequence. Reverse genetics. Based on the knowledge that the disease is monogenic, randomly chosen DNA markers are used until one which co-segregates with the diseased gene is picked up. RNA, a polymer consisting of nucleotides A, G, U and C. Their main types are mRNA, tRNA and iRNA. Satellites or mini-satellites, short sequences of 10 to 60 base pairs repeated 20 to 100 times in a stretch of DNA. These sequences can be amplified by PCR and used to track mutant genes in families. Southern blot, nothing to do with direction. Southern electrophoresis DNA fragments obtained by digestion and discovered a means for identifying the DNA species by this technique. Splicing, removal of introns, non-translatable proteins, portions of mRNA and ligating the exons for the final product. Splicing errors can occur in mRNA resulting in isoforms of the product. Template, a block of DNA sequence in which RNA or DNA is synthesized as the corresponding strand. Transcription, making mRNA from DNA one of the strands. Translation, making peptides through mRNA which provides the amino acid sequence code. Transfer RNA brings in each amino acid for assembly turn by turn. Triplets, the DNA code consisting of 3 bases from A, G, C and T. Western blot, analogs in southern or northern blots, but the proteins which are identified by electrophoresis using antibodies which complex with the peptides in the epitope site. That completes the definition of some of the commonly used terms in the molecular medicine.